Hello everybody, welcome to the second class, right. So, this class we are going to talk about protein structure. As you will see, it will be an introduction to the protein structure without going into too much of details, but some of the some details like amino acid structure and the outcomes of the amino acid structures or the peptide bond, these are the things we are going to look at in some detail, right. But before we go ahead with today's lecture, what we are going to do is we are going to do a brief recap of what we did last class. So, remember in last class we were talking about the protein synthesis on the ribosome, and we were dealing with different aspects of the synthesis. So, we had this the peptide tunnel, that's the figure or the diagram you are seeing out here is belonging to that ribosomal tunnel. This PTC, if you remember, was the peptidyl transferase center, and this is the exit site through which the elongating nascent chain, that is a chain which is being formed in the ribosome, is coming out. Now, not only that, apart from this peptide getting synthesized, or the protein getting synthesized, when this protein or this elongated chain comes out through the tunnel, there are also many interactions it faces, right, or it experiences. Then there is a constriction at some place in the tunnel, right, and we looked at why we have a constriction out there, because of two specific proteins being present there, giving rise to the constriction. And then after it comes out to the exit site, which is a little bit wider, because that is where the tunnel ends. And if you remember, we discussed about the presence of this trigger factor, which was a chaperone, and this trigger factor made sure that the peptide that was coming out, or the amino acids that were coming out, they were well protected from having bad interactions with the surrounding environment. Okay. So, and we also I think talked about one aspect where we said that within this tunnel a question can be asked is, does the protein start having structure or start getting structure inside the tunnel itself? Yes, evidence is there. And as we were discussing in last class, using FRET, people have shown, that means this donor acceptor fluorophores, people have shown that there is development of some sort of secondary structure or even tertiary structure inside the ribosomal tunnel. So, it is not only a place where through which the elongating chain comes out, it is also a place where the protein starts gaining some shape, which possibly helps it later on when it goes to the folded form. Okay. These are some references that I also put up in last class, but I just want to go through these couple of slides really quick. If you have, and hopefully you will be having time, please do go through these references. These most of the materials we talked about have been taken from these references, and to have a much better idea, it is really worthwhile to go through the references. Each and every reference is excellent in its own respect. Okay. Now, let us now move forward and talk about what we hope to discuss in today's class. It is about the levels of protein structure, right. Now, the first question that comes to our mind is, why do we really care about protein structures? Why should we care? Okay. Then this we all know, it is just a repetition of a well known fact that proteins can be considered to be the most versatile macromolecules of the cell. Now, being the most versatile macromolecules of the cell, you can understand that the proteins would be involved in various functions. Typically, each and any function you can think of, and you would see a protein component being there. So, hence protein is so important to us. Then the shape of a protein often directly determines its biological functions, right. What are the types of functions it is going to follow or are going to perform? For example, each and every protein is not going to be able to perform all types of functions. So, there has to be something in the protein structure or in the protein shape itself, which makes it perform the function it is supposed to carry out, right. And hence, the shape of a protein is so important. And if you are going to talk about the shape of a protein, then the shape of a protein comes because of the structure it develops when it finally goes to the native state, right. Then proteins, this is also known fact that there are families of proteins which have similar functions and they have been found to have similar shapes. So, again this is an important point with respect to the structure of the protein or why we should study protein structure. And in general for a protein, remember please focus on these two words in general. What I mean is that in general for a protein to function properly, it has to adopt a very well defined structure, right. That means, if it has to perform a function properly, it better have a structure, a well defined shape. But this does not mean that each and every protein we have within our cells has structure. There are proteins known as unfolded proteins which remain unfolded, they do not have any structure, but even in the unfolded form they are functionally active, and this class of proteins are referred to as intrinsically unstructured proteins or intrinsically disordered proteins. Now, this is a class of proteins we will not be talking about much, but these are also very important because of their flexibility, because they are unstructured. Okay, so, let us look quickly at some major functions of proteins. So, one is binding, we will quickly look at a couple of examples of these. Then is catalysis, that it will help in catalyzing certain reactions, as molecular switches, off and on switches, maintaining structures of cells, that means helping in maintaining the structure, that means as structural components of the cells. Then transport, and when you talk about transport, the protein that first comes to your mind is hemoglobin, right. Okay, it's a it's a 4-mer protein, it's a tetramer. So, what about binding then? Well, here are two examples. The first one is a TA binding protein. Now, this protein you can see, so this is the protein and this is a DNA sequence at the bottom part. So, if you follow the arrow, this protein recognizes a specific DNA sequence or binds to a specific DNA sequence, and this serves as a platform for the initiation of transcription of genetic information. Transcription is so very important, and hence this forms this binding of the TATA of the the TATA, this TATA binding protein. I mean, that is the protein which is binding to this TATA box is the prelude to this transcription initiation. Then we have the well known protein called myoglobin. So, myoglobin is known to bind to oxygen, it is found in muscle tissues, right. And what it does is, if you look at the center, this grayish space filling model, this is a model of the heme cofactor. This has an iron atom in the center where the oxygen comes and binds, right. And myoglobin reversibly binds the oxygen, that means it binds the oxygen and also releases it in times of need. Okay. That is why it said it stores oxygen for use in the muscle tissues, right. So, that means it keeps it there and when it is needed it lets the oxygen go, that is why it is called reversible. Then we have catalysis. The first one is DNA polymerase. So, the DNA replication, we are talking about DNA replication here, this is catalyzed by a specific polymerase that is known as DNA polymerase, right. This copies the genetic material that edits the product for errors in the copy. So, essentially this one helps in proofreading of what you are typing out or what this complex is typing out. Then we have HIV protease. The HIV protease, this replication of the AIDS virus HIV depends on the action of this protein cleaving enzyme called HIV protease. So, this enzyme, therefore, you can understand this enzyme has been the target for many protease inhibitor drugs, hence its significance. Then the switching on and off. So, the RAS group of proteins, these are extremely important in signal transaction or signaling pathways. So, on the left it is called the it is referred to as the off state, and on the right is the on state. So, the left what we see is that it is a GDP bound state which is the off state, and this conformation of the off state significantly differs from the on state where now the RAS is GTP bound. So, you can see if you are trying to figure out where the conformations are different, so follow this arrow of mine, you look at this portion and then you compare the portion out there, you can see it is definitely drastically different. Then you compare these two strands out here and then you also compared this one out here, again this is different. So, that means when GDP and GTP are bound, then the protein is respectively in its on and off conformations, which accordingly is sensed and then transfers signal through the signaling pathway by combination with other proteins. Okay. That is why it says the difference causes the two states to be recognized by different proteins in signal transduction pathways. And last but not least, we have the structural proteins. One is the silk. You have heard about silk. Silk derives its strength and flexibility from its structure. It is a giant stack of anti-parallel beta sheets, as written out here. Its strength comes from the covalent and hydrogen bonds within each sheet. The flexibility comes from the van der Waals interactions. And then we have the actin fibers, which are so important for muscle contraction and the cytoskeleton. So, these are structural proteins. They give structural balance, or they help the cell in the structural aspect. Okay. Now, having looked at some different functions, right, I mean, I am not saying that these are all the functions that we have been able to cover, but at least these come under four very broad classes. The other one I did not show was the transport, and I said that hemoglobin does do that, right, we all know that. So, whatever the different levels of the hierarchy of protein structure. So, all of us know that there are 4 levels of protein structure. I am sure you have all of you have done some preliminary course in biochemistry. So, we will not be going through this in very details, but you can see the first one, which is the primary structure. This is essentially your amino acid sequence, right. It is the sequence or the way in which the amino acids are placed in the peptide chain, or way it is synthesized by the ribosome. Then you have the secondary structure. This is mostly your alpha helix, beta sheet, or a combination of these. Then you have the tertiary structure, which, what is the tertiary structure? It is essentially a packing of your different secondary structural elements. And then you have your quaternary structure. The quaternary structure itself is like a packing of your tertiary structural elements, right. So, these are the 4 levels of protein structure, and this is what is referred to as the hierarchy. So, that means you have the primary first, then the secondary, then the tertiary, and then the quaternary. Okay. Let us start looking at the amino acids. What are these amino acids? The amino acids are so essential for us. Why? Because these are the building blocks of proteins. So, let us look at the amino acids, the structure. So, what we have is, here we have, it is an amino acid, right. So, we have an amine group out here. So, here it is protonated, NH3 plus, then it is an acid. So, we have a carboxylic acid out here, here it is deprotonated. Now, we have a side chain which is R, and here we have a carbon atom, right. Okay. So, this carbon atom is very important. It is called the alpha carbon atom, and this alpha carbon you will see, we will mention it quite often as we go on through today's class. Now, this R is a side chain, and depending upon the R, your nature of amino acid would vary. That means, whenever you have a different R, you have a different amino acid, and that is how you have 20 different amino acids. That will just depend upon the R, right. That is what it says in the bottom. There are 20 different amino acids, and these are L amino acids. These are the naturally occurring 20 different amino acids. Okay. So, depending upon the nature of the R also, the nature of the amino acid will change. Nature means whether it is polar, whether it is non-polar, whether it is charged, neutral, all will depend upon this. So, what you can understand is, this is the basic structure of the amino acid, and here what you have is, each amino acid has this amino group, the carboxyl group, an R group, and which gives itself the identity, and then you have the C alpha in the middle. This is essentially your chiral center. Okay. Now, what do we have on the right? So, here this one, what we are seeing here are the torsion angles. So, what we mean by torsion angles is, if you look at this N C bond or the C C bond. So, this N bond, this C is called the C alpha. There is a C alpha here. So, this C alpha and this N, right. So, this is referred to as the N C bond. Okay. Then this C alpha, then this C alpha, and this C, this is the C C bond. So, if you have a rotation around the N C bond, this rotation, this torsion angle is referred to as phi, and if a torsion angle, along the C C bond, this is referred to as psi. And then this C n or this C n, which you see, it is kind of looking like a plane, it is shaded like a plane. This is your peptide bond. So, this one is a peptide bond. We will look at it in the next slide in a little more detail, and any torsion around that ball is represented by the angle omega. So, why we are looking at it right now? Well, possibly it is the best place to look at, because we have just started looking at the amino acid structure, because this leads to one of the very famous plots in biology or biochemistry. It is referred to as the Ramachandran plot, okay, and we will come to it later. So, what we will mainly be focusing on are these two angles, the phi and the psi, right. Okay. So, let us look at the different types of amino acids. There is the natures, and as I said, how are these natures different? This nature depends upon the type of R you have. So, if it is hydrophobic amino acids, then you can understand your R is essentially hydrophobic in nature. So, let us look at the hydrophobic amino acids. So, you can see the first one is alanine. What is R? CH3. Then you have valine, it becomes more hydrophobic. So, this is the group. Then you have phenylalanine, then proline, then isoleucine, leucine, and methionine, right. So, this is your hydrophobic amino acids, or the class, or the the different amino acids coming under the class of hydrophobic amino acids. Then we have the charged amino acid, that means the R group is charged out here. So, we have aspartic acid, we have glutamic acid, we have lysine, and we have arginine. This charge acid, again these charged amino acids mean that the respective side chains or the R groups, they have charges. Then we have the polar amino acids. The polar amino acids, so is serine. So, these are not charged, but these are polar, okay. So, neutral, but they are polar. Then you have threonine, then you have tyrosine, then you have histidine, then you have cysteine, asparagine, glutamine, tryptophan, okay. So, then what we have is we have three different classes of amino acids. One is hydrophobic, one is charged, and then we have the polar amino acids. Let us look at the peptide bond formation. So, what do we have is, how does the peptide bond form? Obviously, to have a peptide bond form, we will need two amino acid units, right. So, one amino acid A, say one amino acid B, or it may be the same amino acid A and A, they come together and they form the peptide bond. So, this is how it happens. So, you can see out here. So, this is one, it is coming like this on your left, this amino acid is approaching from your right. Here the N group then combines or reacts essentially with the C group out here, this C atom, there is a loss of water molecule, and then you form the peptide bond, right. So, that is why it is a reversible arrow. So, if you lose, you form the peptide bond, then if you are trying to hydrolyze it, it will go the other way. But it does not mean that if you put a protein in water, it will readily get hydrolyzed. It just tells you what goes on. Then, when you are forming this peptide bond, what you get, if you look at very carefully, is from the structure is, on one side you have this amino group, and the other side you have the carboxylic acid group, right. So, this amino group comes from the amino acid to your left, and this carboxylic acid group comes from the amino acid to your right. So, the amino acid to your right has the chain as a side chain R2, and the amino acid to your left has the side chain R1. That means, if R1 and R2 are different, then obviously these are different amino acids. So, then to the left you have the N terminus, and then to your right you have the C terminus or the carboxyl terminus. The C terminus because it ends with the COH group, the N terminus because it starts with the NH2 group or the mean group. So, that is why this is referred to as the N terminus, and this is referred to as the carboxyl or the C terminus. Now, these are some terminologies you have to be familiar with throughout this course, because we will use it often. Okay. Now, what about an extended polypeptide chain? So, what do we mean extended polypeptide chain? Here what we saw is we just had two amino acids coming together, the main group of one reacting with the carboxyl center of the other one, loss of water molecule, we form a peptide bond. Okay. Now, when we get a protein, right, when we get a protein like myoglobin, say, has 153 amino acids, that means you can understand there are 153 amino acids which are forming, which are coming together and being a part of the molecule, forming the peptide bonds. So, this is typically how a peptide bond looks like, right. You can see you have 1, 2, 3, 4 different amino acid subunits, are the R groups R1, R2, R3, R4. These again give specific identities to the proteins or to the amino acids we are combining. Okay. So, here this dotted line, if you look at this dotted line, or before going to the dotted line, if you look at these shaded regions, right, as it says R stands for the different amino acid side chains, then the peptide bond is represented with a shaded region. So, this shaded region is which the shaded region is essentially a plane which contains the peptide bond. You can see here also, here and here. So, there are 3 peptide bonds here, and this the last amino acid is making another peptide bond. Now, what you can also see is this CO and N, this N is in blue, O is in red. Throughout, what happens is here is a dotted line. What it means is this CN has a certain double bond character, and because so this it is a symbol of delocalization. Electrons are delocalized. So, if you remember, there was this omega torsion angle around this CN bond, and this omega is important because the CN being having partial double bond character, it is very hard to rotate around, and this gives rise to many cis and trans configurations you will just see. Okay. And then what are these arrows? These arrows essentially tell you your dipole moment vectors. Okay. So, taking a closer look at the peptide bond, let us look at it again. So, here we have the C alpha, I told you it is very important, then the C, this is N, again the shaded region is your peptide bond, or this is the plane which contains a peptide bond, plane because the peptide bond essentially is planar, as we just talked about, it has double bond character. So, some specifics, there are 6 atoms lying in a single plane. So, you can understand C alpha, C, N, C alpha, these make 4, then H, then O, 6. There are 6 atoms lying in the single plane. Now, the bond between C and N has double bond character. So, this bond we are talking about C and N, this is the one which has double bond character, right, and this is a trans configuration. So, you can see this part and this part. So, if this is the one which is having the double bond character, so this is on one side of the double bond, this is on the other side of the double bond. So, this is a trans configuration. Now, whatever rotation being permitted, so rotation or the flexibility, so rotation is permitted between the N C alpha and the C alpha C bond. So, what I mean with this is the N C alpha, this is the N C alpha, you can see this is a single bond, rotation is permitted around it. Then you also have the C alpha and C again. So, these were the different angles we were talking about. I will come to this come to that again, and this C alpha C, again this is a single bond, rotation is possible around this bond. Okay. Now, once we had talked about the trans configuration which was in the previous slide, then the other configuration obviously is your cis configuration. Now, you can see this again being the amide plane, we have the same 6 carbon atoms on the plane, but now what you have done is you have taken the trans configuration and brought this. So, you can see, so if you go back, you can see these two were on different sides of the bond. Now, you have taken this and brought it to the same side, and that is what you have. This is your cis conformation of the peptide group. Okay. So, let us look at some details into the torsion angles. So, what do you mean by the torsion angles? So, this is what we mean. So, what we have out here is, if you look at it carefully, is, so this is the N, this is the C, this is one amide plane, again this is another amide plane. Okay. Now, what we have is, this is the alpha carbon. Remember, I will come back to this alpha carbon as I told you many times. Now, this alpha carbon is very important because this is like a hinge, this is like a pivot point around which both these planes can rotate. So, you understand. So, this one is restricted, this guy is restricted too, because this CN, peptide bonds, double bond characters cannot freely rotate. However, this plane and this plane, they are hinged at one point, which is your C alpha, and along this C alpha they can freely rotate. How? This C alpha and this guy, this C, this C alpha and this atom which is N, around these are single bonds, right. So, around these the planes can rotate. Okay. So, then any rotation around the C alpha and N, this bond, there is a C alpha and N, please follow my arrow, this is referred to as the phi angle or the phi torsion angle, and any rotation around the C alpha and C, this one is referred to as the psi torsion angle, and that is what this one says. The N C alpha bond is your phi, and the C alpha C bond is referred to as the psi torsion angles. And if you have, I am sure you already know, these two in combination, if you plot, it gives rise to the Ramachandran plot, and it actually tells you what are the possible or admissible psi and phi values that a protein or a polypeptide chain can take. Okay. So, again this is the peptide bond, you are seeing the same thing, only just from a different angle. You have this phi along N C alpha, you have this psi along C alpha C, and it just this 1.45 angles, this just tells you the different bond lengths involved. So, here in the last in this slide you had 2 planes. Now, what you are seeing is you are having 3 planes, that means 3 peptide bonds, 3 planes having this peptide bonds, and again you are looking at the torsion angles, right. Okay. So, let us now go into a little more details. How do we visualize the torsion angles? So, let us try to look at it. Okay. So, what we have is here again we have the extended chain, these dotted lines they constitute the planes. The planes are those planes where you have the amide bonds housed or amide bonds located. Okay. Now, this is your phi, this is your C alpha, please again follow arrow or the pointer, this is your C alpha, then this is the C and this is the N. So, any rotation around this C alpha N, any rotation on the C alpha N is referred to as the phi, any rotation on the C alpha C will be referred to as the psi. Now, how would you look at it? So, what it says is if you are having a chemistry background, you will realize this word Newman projection, you are trying to project it. Okay. So, when you are trying to project it, how do we look at these bonds around which the torsion or around which we are rotating. So, if you are going to look at this phi angle, what we do is we look at the N C alpha, right, that means the C alpha stays at the back, right, and here we have the N out here. Okay. Now, so this is the arrow, this is how we are looking at. Now, if you are looking at the psi angle, this is what we go. So, you look at this is the arrow, we look at it from this side, we look at the carbonyl first, this carbonyl carbon first, and then we look at the C alpha at the back. Okay. So, the C alpha is always at the back here. Okay. Now, here what we see is, so this is to the left of your screen, we are looking at the phi angle, try to visualize it a little bit more. So, again go back to this arrow, this is what we are looking at, we are looking along the N C alpha bond. So, here this is the N, okay, this is the N, this is the same N as this, and then this is the C alpha, this big circle. So, the N represents that point of N, and the C alpha is this big circle, right. That is how a Newman projection is drawn. Okay. So, you are now looking along the line, right. Now, once you are looking along the line, now see what happens is, if you look at this N C carbonyl and this carbonyl, you can see they are on opposite sides, right. So, this one is pointing on one side, this guy is pointing on the other side, and this is referred to as the trans conformation. Remember the trans conformation where the COs are pointing on the 2 sides. Okay. So, this is what you are looking at. Now, what happens? Now, do not worry of this positive sign right now. So, now we start turning, that means we start moving around this N C alpha bond. Okay. Now, you can see we are moving, we are doing a torsion, that means we are moving. So, this is a plus 100 degrees, do not worry about the plus I will come to the convention of what is plus, what is minus. Okay. So, here you can see when we have moved this, so the angle between this guy and this guy is now 100 degrees. So, initially what I said, the angle between this one and this one is 180. Now, the angle between this one and this one, as this arrow is showing, is 100 degrees. So, this is how you look at your phi, right, and these are the types of changes that are happening when you are moving around the C or N C alpha bond. Now, what about the psi? Now, again we do the same thing, we look at it along this bond, right, along the C alpha bond. Okay. So, when we look at it along the C alpha bond, okay, so this is what we have. So, this is the C, again this is the trans, because here what you see, this is the NH. When we are saying trans, here we are talking about the NH values. Now, if you go back to the phi, when we were talking about the trans, see, in this case the trans were about the COs, right, the CO is pointing in this direction, the CO is pointing in this direction. But when we are talking about the psi, so that was about phi, but when we are talking about the psi, now the trans is with respect to the NH bond. So, this is NH is pointing in this direction, this NH is pointing in this direction. So, again this is the trans configuration, you can see this is the farthest apart, and then you do a minus 60 degree rotation. Okay. So, you are rotating like this, you do a minus 60 degree rotation, you can see like this, and what you get is this one coming closer to this. Okay. So, this is how a rotation would be done or a torsion would be done if you are doing, or if you are moving, or if you are rotating along the C alpha bond. Okay. So, this is one of the steps of trying to visualize it. Okay. Let us look at some better representations. Okay. Let us go forward. Let us pick torsion angle phi. Now, how do we look at it? Okay. So, these are some conventions. This angle is measured by looking along the bond with the nitrogen atom in front and the alpha carbon to the rear. Okay. Now, remember, what does it say? It says that if you are looking at this bond, if you are trying to see what this phi is, then you would rather look through the nitrogen, right, that means through the NC bond with the nitrogen atom in front and the alpha carbon and back. This is exactly what we saw here, right. N is in front, C alpha, it is back. Good. Now, what is generally measured? It is measured from minus 80 to minus 180 to plus 180, right, full rotation, with the positive direction being when you turn the rear group clockwise. So, that the rear carbonyl bond is clockwise of the front carbonyl bond. Okay. Again, so this you see the range of measurements that can be done now, or the range of angles that it can take. Now, with the positive direction being defined as what? When you turn the rear group clockwise, so that the rear carbonyl bond is clockwise of the front carbonyl bond. Now, let us go back. So, this is what I said. So, the rear carbonyl bond. So, this is the rear bond. So, this is the rear carbonyl bond. So, that means this rear carbonyl bond is clockwise with respect to the front carbonyl bond. The front one is the one which is here attached to N, and the rear one is the rear one here attached to C alpha. Okay. So, this C this C is your rear one, which is C coming out from C alpha, and this is directly attached to N. So, this is your front, this is your rear. So, what this one is saying is that you move it clockwise. When you move it clockwise, that means when the rear carbonyl bond is always clockwise of the front carbonyl bond. Okay. So, that means again, so this is the real carbonyl, you can see it is clockwise in front of, it is clockwise of the front carbonyl. Here also the rear carbonyl, it is clockwise of the front carbonyl. Okay. So, that is why it is called plus 100, because that is the angle, but then this is how this is the convention of giving it the positive sign, right. Or when you turn the front group counterclockwise, so the rear carbonyl bond is always clockwise of the front. Okay. So, these two mean the same, right. Either you start tweaking the rear carbonyl or you start tweaking the front carbonyl. The rear carbonyl you tweak in a clockwise fashion, and to get the same effect with the front carbonyl you rotate it in the anti-clockwise fashion or the counterclockwise fashion. So, this is what I am saying. So, this is your rear carbonyl, right, or this is your rear carbonyl. You can see this is your rear carbonyl. Now, if I have to move it in this direction, this direction would be counterclockwise, right, this direction would be clockwise. So, for the front carbonyl, this direction is counterclockwise, and that is what we have done here, we have moved out here. So, that is the convention that is given to it. Okay. So, that is your positive direction. So, positive in a nutshell for a phi torsion angle is where your rear carbonyl is always clockwise or in a clockwise orientation of your front carbonyl, right. Keep that in mind, please. Okay. So, now let us look at some angular orientations. So, what we are doing is here this is, so these are different representations. This is phi is equal to 0, phi 180, then phi 90, phi 90. How do we look at it? Okay. So, here you can see this is N, this is the C alpha, right, this is exactly the same thing we are looking at, right. This is where phi is 0. So, why is phi 0? You can see why phi is 0, because CO they are top of each other, same direction, right, and so this is what you are looking at. Okay. Now, what happens is you start rotating. What do you do is you can see what you have done is you have rotated one of these. Okay. So, this see this N C O, this is out here, but this one you have rotated, the C C O you have rotated, you can see. So, that means that this is the back, this is the back N C O you are seeing out here. So, if you look at this, these N C O and the C alpha C O, these are given, these are represented by two lines. The first line is the N C O, the back line is the C alpha C O. Now, what you are doing is you are doing a phi 180. So, when you are doing a phi 180, what you are doing is here what you see is you have the N C, okay, N C O, this one has come like this. So, that means clockwise you have done it phi 180 degree clockwise like this, okay, and that is why it has come here. So, see you have given it a clockwise flip, right. Then what can be the other one of looking at? So, let us see out here. So, again this is the NCO, right. So, this is NCO, this is the C alpha CO. Now, this is we are talking of 90 degrees, the same thing happens here. This is NCO and this is C alpha CO. So, again here you can see this we are going like this, right. You can see the direction, it is a positive Y, because this back carbon is always clockwise with respect to your front carbonyl. Now, look at this, what has happened here is your back carbonyl is what? Your back carbonyl is behind your front carbonyl, is not it, by 90 degrees. So, this is a 90 degree in the other direction. So, it is essentially negative, right. So, this is a positive direction, that is how it is rotated, but this one is rotated the other way like this. So, there is a negative direction. So, what you do is, this is a very good way of looking at it. You just keep in mind the way this stick or this line is being moved around. Okay. So, if it is moving around like this with the C coming like this, it is phi is equal to 180. Again you move the stick in this direction again. So, this is clockwise with respect to this, right, it is again positive 90. However, here you move this back stick anti-clockwise, that is counterclockwise, then it is negative, because positive by convention is what, where the C alpha CO is clockwise to your NCO. Clear? Please go through it again, think about it with a very cool head. This is an excellent representation of the same. The phi angle, this is as seen along the NC alpha axis, that is what we are looking at, and this we have kept psi as 180 degrees. Okay. Now, then once we have talked about phi, now let us talk about psi. What are the torsion angles psi? So, this angle is measured by looking along that bond with the carbon of the carbonyl group in the rear and alpha carbon to the front. Okay. So, that means with the carbon of the carbon group in the rear, it does not matter how you look at it, there I think we had the other way around. Now, measure from minus 180 to plus 180 with the positive direction being when you turn the rear group clockwise. So, the rear nitrogen bond is clockwise of the front nitrogen bond. Okay. So, remember in that case we talked about the carbonyl bond or the carbonyl group. So, here what we are talking about? We are talking about the nitrogen NH. So, here it says what is the positive direction or what is your definition of positive? So, when you turn the rear group clockwise, so that the rear nitrogen bond is clockwise of the front nitrogen bond, right, or when you turn the front group counterclockwise, so that the rear nitrogen bond is clockwise of the front one. Okay. So, let us try to take a quick look at this. So, here it says the positive direction being when you turn the rear group clockwise, so that the rear nitrogen is clockwise of the front nitrogen bond. So, here let us look at this representation again. So, this is a C alpha, right. So, this is a C alpha and this is the carbonyl. Okay. That is what we have said, that is the convention we are going to follow. Okay. So, now what you see is here you can see what has happened is I have taken this and go back and talk about it with the positive direction being when you turn the rear group clockwise, so the rear nitrogen bond is clockwise of the front nitrogen bond. Okay. Let us see. So, here again you see these stick representations or line representations. The first one again is this C alpha n and then the back one is this C n, right. Now, what you are doing is you are doing a positive flip. If you are doing a positive flip, remember this n at the back end has to be clockwise with respect in a clockwise orientation with respect to your front end. This is what exactly I have done. You have rotated it like this. Okay. That psi is equal to 180. Okay. See, it says with the positive direction in being when you turn the rear group clockwise. So, you are turning the rear group clockwise. So, the rear nitrogen bond is clockwise of the front nitrogen bond. Okay. That is what you have. So, this is now in clockwise of the front nitrogen bond. Okay. Now, how do we define 90 now? Again we do the same thing out there. So, this is how we are flipping. So, this rear group you see now comes here, again it is clockwise in front. Okay. But what is negative 90, right, that means doing a minus 90, then what happens is you are taking the rear group and just flipping in the other direction. Okay. And because you flip it in the other direction, then it is essentially going the other way, right. So, this is what we were trying to also say out here. Okay. This is what this minus 60 represented, is not it. So, the minus 60 represents what? Remember, this is your C alpha C. So, actually we have it the other way around, the C should have at the back side, the C alpha should have come in front, that is why it is given a minus 60. Please try to go back, try to take a look at it and realize why this is minus 60. However, I think we have had a decent idea of these torsion angles, how to take a look at these. Okay. So, here what we are doing is we have looked at the C alpha C bond and we have kept phi is equal to 180 in this case. Like in the last one, we were looking at this phi, we kept psi is equal to 180. Okay. Right. So, then a couple more examples. We take an amino acid, right, here phi is equal to 180, the psi, we are looking at the psi angle. So, this is psi is equal to 0, when psi is equal to 0, remember psi is equal to 0, the NHs were on top of each other, and when psi is equal to 180, so they are away from each other, that is what you are seeing out here, right. Same thing out here, now you are looking at the phi, this is phi is equal to 180, when you phi is equal to 180, remember when phi equal to 180, the C's are pointing away from each other, and when phi is equal to 0, you can see the C's are almost pointing in the same direction. Okay. Again here psi has been kept at 180. So, this is when you think about this torsion angle, this is what you should always think about, that you are rotating against one bond or you are rotating against the other bond, you can keep the other one fixed, and then what you do is you try to look at different orientations, that means a certain value of psi, a certain value of phi, and that is how you get or give rise to the Ramachandran plot. Okay. So, then we have looked at these aspects of what the phi and psi mean, or how we can take a closer look at them. So, let us look at some unfavorable combinations. So, this is what we have, you can see what are these values, this is phi is equal to 0, psi is equal to 180, right, and what has happened here? What has happened here is you can see the CO and the CO, they are very close. So, it is non bonded contact, high energy, it will be unfavorable. Okay. Then, so that is why it is unfavorable. Then you look at this again, now phi is equal to 180, psi is equal to 0, what are you having? See, remember when we are talking about phi, keeping the psi fixed, when we are talking about the phi, we are always talking about the carbonyl groups, looking at the orientation of the carbonyl groups. When we are talking about the psi keeping phi fixed, we are talking about the orientation of the NH groups, is not it. Now, here also NH have come very close to each other, because psi is equal to 0, non bonded interaction, repulsion, high energy, hence this is unfavorable, right. Okay. Let us go to the last one, here psi is equal to 0, phi is equal to 0, psi is equal to 0. So, what do you have? You have huge amounts of interactions out here, and because you have huge amount of interactions out, again this is not favorable. So, what you can see out in between, there is a third one. So, this is phi is equal to minus 60, psi is equal to 180, where you see here the amount of non bonded interaction, that is why you do not see these big spheres, these big spheres represent the non bonded interactions, the repulsive interactions you are having. That means, you are having this steric congestion or steric repulsions, but here you do not see those. That means, this is the one which is possibly one favorable conformation for a particular protein. Okay. Remember, this depends upon the proteins, right, or the secondary structures these amino acids take. Okay. What did we do all this exercise for? We did this exercise because we want to look at the Ramachandran plot, and this is what the Ramachandran plot looks like. So, you can see it is a plot of psi against phi, both being in degrees, right. Okay. So, this area is essentially dominated by your helix, this is your extended chain, this area it is a big one, it is dominated by your beta type of secondary structures, this was alpha helix, and this is your another alpha helix, but can you tell me what this one belongs to? Yeah, you are right, it is the left handed alpha helix, or alpha is a right handed, that is the normal one we look into. So, some quick but significant aspects of these. So, the red regions are those combinations of the backbone torsion angles phi and psi that are allowed, because they do not give rise to any steric hindrance. So, this is the figure we are talking about, right, And so, this is the red bonds are the bonds which are really allowed, favorable, no steric clashes. The pink regions are allowed if some relaxation of steric hindrance is permitted, that means they are partially allowed. Here you have to relax your steric requirements a little bit. The commonly occurring protein structural elements are marked at the positions of the average phi and psi values. We will go back to it again. And on the right, the shaded region we have, it belongs to a left-handed helix, which is only rarely observed in short segments in proteins. So, it is only short segments of proteins do you really observe it, otherwise it is not that common. So, then going back, we take a look at it again. You can see this is the beta, this is the left hand alpha helix, this is the right hand alpha helix, this is extended chain. Now, here itself you have different regions, right. Here you can see these different circles. See, each and every point on this corresponds to a definite psi and a definite phi. And you look at these values, this psi goes from plus 180 to minus 180, this also goes from minus 180 to plus 180, right. You can see for all these, the only positive values of phi we have is mainly for the left hand alpha helix. Otherwise, for all of these, essentially phi is always in the negative, right. We go ahead and take a closer look. So, these are the phi and psi regions of different secondary structures on the Ramachandran plot. This is an another figure of the same thing. Okay. So, this kind of tells you a little more detail. We had everything written down there. So, this is your alpha L, which is the left hand alpha helix. Now, this is your regular alpha helix, which is right handed. This is a pi helix, it is a type of helix. This is like a 3/10 of a helix, that is why it is called 3. This is your right hand alpha helix. Then this was your helical region, right. What do we have in the beta region? What we can see is, you can see this one arrow pointing up and one arrow pointing down. These two, I should have had made it a little bigger, but if you kind of strain your eyes, what you will see is these two are pointing in different directions, right, and hence this is an antiparallel beta sheet. So, this is the region where the antiparallel beta sheet phi and psi angles come in. So, this you can see now these two arrows are pointing in the same direction. So, then this is what is referred to as a parallel beta sheet. So, this is the antiparallel, these two arrows pointing in different directions, this is the parallel beta sheet, both in the same direction. This is where they come in, right. Then what we have is here the C, this is the collagen triple helix. We will come to this arrangement of helices later. So, these are in a triple helix, these are the psi average psi and phi angles that the amino acids take in the triple collagen helix, on the collagen triple helix rather. Okay. And to end this discussion about the structures of amino acids, let us look at the Ramachandran plot of glycine. Now, why is glycine so important? Remember glycine, the R, what is R? R was H for glycine, was not it? So, think about the extent of freedom it will have. So, you can see throughout the phi and the psi and phi plane or the psi and phi plot, you have these glycine values. That means, glycine can or has the ability to what? Spread around a range or cover a range of psi and phi values. Now, that is why glycine is so unique, because it is so flexible. It just straight away comes from the flexibility. So, then in a nutshell, what is the Ramachandran plot? The Ramachandran plot, then in a nutshell, what it does is it tells you this. It tells you is it gives you a plot on the phi and psi angles, right, and it tells you, okay, I cannot have all the possible psi and phi angles. Why? Because as you saw in the previous slide, that means the slide previous to this, there would be combinations where the steric congestion would be so high, the non-boundary interactions would be so high that the structure would be very unstable, and hence it would be very high energy and would seldom be populated. Remember I mean, if you think about the Boltzmann distribution, and hence those regions would not be occupied. Those regions in the phi psi angle or the psi phi plot would not be occupied. Instead, we only have to consider on these regions, on these shaded regions, because these are the ones which are covered by the different secondary structural elements in the different proteins, whether it is beta, alpha or mixtures of those, right. I think this was what I needed to discuss with you about the Ramachandran plot. See, we do not think too much of the Ramachandran plot, but if you kind of try to think about it, or if you kind of try to dwell on it for some time, if you have the psi and phi angles, you essentially have the conformation of the protein, and that is exactly what G. N. Ramachandran did. Okay. Now, let us take a first step towards protein structure. Initially, I mean, we have looked at the building blocks, we have looked at the Ramachandran plot, we have looked at the hierarchy of structure, but this is a unique thing that we are going to look at right now. Okay. The first question is, and I am sure which has come to your mind, is what determines the protein fold. That means, whether the final protein is like myoglobin, which is fully alpha helical, or like lysozyme, which has mixed alpha and beta, or some beta sheet protein. How does it know? How does the protein know what structure, what native structure it has to take? Okay. So, Chris and Finson tried to, and his co-workers and some other groups tried to address this problem. So, he did a very simple experiment. What he said is, he took a folded protein, he unfolded it, it went to the unfolded protein, you got the unfolded form of the protein, right, and he did a dialysis, and then what he saw is that he got the folded protein back with almost the same activity. Now, what activity I am talking about, I will tell you within a very short time, just hold on. This is Chris Edfinson. So, he was awarded the Nobel Prize for his pioneering work in 1972, and what he said is, this is his paradigm. It says that the information required for correct folding of the protein is contained within the amino acid sequence. What it means is that it is a primary sequence. See, it is a primary sequence which is the most basic, that is why it is called primary sequence. It is a primary sequence where that information is encoded, that what type of structure the protein should be going for once it is folded with all its tertiary quaternary contacts. Okay. So, how did it do it? So, what it did was, he took this 124 residue protein, bovine pancreatic ribonuclease, right, also called RNase. So, this one has 4 disulfide bonds, that means 8 cysteine groups. You can see the different groups are out here. So, for example, if we start from say 1 out here, this is, so here is one group, here is one, here is one, and here is one. So, they are about like this 4 disulfide bonds. Okay. Now, what did he do with this? Why is this disulfide bond so special? Right. So, this is what he did. So, what he did was, he took this native ribonuclease. Remember, these lines are essentially your 4, you can see these 4 lines, these are your 4 disulfide bonds, and he added reducing agent. So, once he added reducing agent, what happened was, the reducing agent reduced these disulfide bonds and converted the SS bonds to SH. That means, the disulfide bonds do not exist. So, now the protein is a little more free, because you can see these contacts no longer exist, and the principle is now he denatured the protein. That means, he added either heated or he added urea. So, he added urea in this case, and then he unfolded the protein. Okay. So, that means, this disulfide bond you can see out here, this was reduced with the reducing agent. Okay. This was reduced with the reducing agent, and once he reached this reduced ribonuclease, he unfolded the ribonuclease with the urea, and then when he removed the denaturant, he saw that it went back to the reduced ribonuclease. Then what did he do? Okay. But this was a scheme. The scheme was essentially that he started from the native, which is having all the 4 disulfide bonds. He reduces the disulfide bonds, he adds a denaturant urea, he unfolds the protein, then he takes the urea out and he does something, and he sees what happens. Okay. Let us look at his 2 different experiments. Okay. The first observation. So, this is the native form. The native form means the one which is found, which is 100% active, right. See, it is ribonuclease, that means it is an enzyme. So, it will be having enzymatic activity. So, it is good, because then you can, whether it has attained its native structure or not, how would you know? You would know by its activity, right. Okay. So, this guy is 100% active. Then you look at this arrow. So, this is first experiment, he reduces it. Okay. he reduces it by a reducing agent, say beta mercaptoethanol, then he adds 8 molar urea, chemical denaturer. So, you can see all these are, you have these 1, 2, 3, 4, 5, 6, 7, 8, right, these 8 SH groups. Now, what it does is he removes the urea. Okay, so, it says denatured, inactive, that means now the protein is denatured, it does not have the active site intact, and hence it cannot act as an enzyme. So, it is fully inactive. Now, what it does is he removes urea. Okay. So, once he removes urea, then he oxidizes. Once he oxidizes, now what happens is then he gets back this 90% active state. So, what it tells you is that he removes urea, he oxidizes, and the protein, he waits for it, and the protein automatically goes back to its 90% active state. Okay. Almost 90%, I mean almost 100%, greater than 90%. So, the first one is this, there are 4 SS bonds reduced using mercaptoethanol, and the protein was denatured 8 molar urea. The enzyme was thus rendered inactive. On removal of urea and the reducing agent through dialysis, the protein was found to slowly regain its enzymatic activity. This led him to the thermodynamic hypothesis. I will come to it later, what I mean by the thermodynamic hypothesis. Okay. So, just a couple more slides before I end. Now, what was the other experiment? Now, look at the other experiment. He takes the native form. Okay. Again, the first step is essentially the same, right. He reduces it, then he denatures it. So, it is a denatured. Okay. Now, what it does is, you look at this step, he removes urea, then oxidizes, but here he does a different thing. He first oxidizes and then he removes. Now, because now he oxidizes before removing urea, the protein is in a denatured state. So, now it is actually a double bond, scrambled. It cannot form all these native bonds all the time. Maybe a small percentage would form, but mostly it would be scrambled. That means, you would be having different cross links. That means, say for example, this SH might cross link with this SH, this SH might cross this SH, and so on. Now, what he found was, after oxidizing, when he removed urea, that means after he has formed these scrambled disulfide bonds, when he has removed urea, he does not get activity. It is only 1 to 2% active. That means, maybe only 1 to 2% of these molecules actually gained the native structure where it had the correct disulfide bonds, right. That is why the activity is so low. So, then the steps of his second experiment, it is called the disulfide scrambling. The 4 SS bonds were reduced. This was the first step, and the protein was denatured. The fully reduced protein was allowed to reoxidize under the denaturing conditions. Okay. So, initially we removed urea and then we allowed it to oxidize, but now we allow it to oxidize and then we remove urea. So, now urea was removed and the protein was checked for any enzymatic activity. Very low activity on the enzyme was observed, that is, enzyme is essentially inactive. However, if the scrambled protein is now exposed to small amounts of mercaptoethanol, so this is a very neat experiment, but a brilliant idea. So, what he now does is, he has a scrambled protein, right. Now, if the protein has to rearrange, remember the protein can rearrange, right. So, that means you have to break the disulfide bonds. So, what he now does is, he just exposes it to small amounts of disulfide bonds, I mean a small amount of reducing in this beta mercaptoethanol, and the disulfide indices now takes place and the protein regains its activity. Now, this is really nice, this is really important. What message does it deliver? This is what the thermodynamic hypothesis says. So, this is essentially the last slide. The 3-dimensional structure of a native protein in its normal physiological milieu, that solvent, pH, ionic strength, presence of other components such as metal ions or prosthetic groups, temperature, etc., its normal physiological environment is the one in which the Gibbs free energy of the whole system is the lowest, right. That is the most stable. That is, that the native conformation is determined by the totality of interatomic interactions and hence by the amino acid sequence in a given environment. Okay, sorry. So, hence by the amino acid sequence in a given environment. So, in a nutshell, what is telling is that, okay, going back to his second experiment, when I oxidized this before removing the urea, I found the scrambled disulfide bonds, but the moment after I removed the urea, it was essentially inactive, but only 1 to 2% activity. But the moment I allowed it to rearrange, that means I added the small amount of reducing agent for it to rearrange, then the protein immediately rearranged. Now, immediately I am not talking about the kinetics here. So, okay, do not quote me on that. That means the protein did rearrange over a function of time until it gained almost that 90% to 100% activity. What does it mean? That means the protein actually went to a thermodynamically more stable state when it was allowed to. And this is what it says, by the totality of interatomic interactions and hence by the amino acid sequence. But that is, there is the amino acid sequence which defines the totality of the interatomic interactions and which finally determines what structure at the end the protein is going to have. Okay. So, this is the Nobel plaque that was given to Christian B. and Finson in 1972 for his pioneering work. We will stop here. What we will do is, the next class we will start talking about the secondary structure, and I just want to end this class with a request that you guys please take a close look at this Ramachandran plot, look at the phi and psi angles and try to understand the importance, try to understand the different non-boundary interactions you guys that take place, and why we have that only those small regions in the Ramachandran plot which the proteins in different conformations actually cover. Okay? Thank you.